Hey everyone. Could you please give me a feedback that can you hear me? Thank you. Thank you. Good. Okay. So let's start the uh, workshop about gene editing. Special thanks for Parvati School in clinical uh, biology. And yeah, it's about gene editing or genome editing. Um, as you may know, um, genome editing is a precise system in specific and targeted gene manipulation. The main aims of the system is, to study gene function and also biological mechanisms and study about pathological diseases. Uh, so, what's the abilities in about genome editing? Uh, the first one is uh, targeted mutagenesis, indel mutagenesis. Indel is uh, the abbreviation of two type of mutagenesis, insertion and deletion. And the next one is suppressing and activating gene expression. And the last one, the last ability of the genome editing is on, chromosome rearrangement. Okay. Uh, let's take a look on the mechanism of the genome editing. Is two main process. The first one is creating a DNA break, and the second one is repairing the break. There is two main cellular pathways to repair the break. Uh, the first one is homology-directed repair, or HDR, and the second one is non-homologous and joining, or NHEJ. Uh, this system, this this one is homology-directed repair. Uh, it's you know when you make the uh, create the break in the DNA, uh, you need uh, a donor fragment of DNA that helps to the system, the repair system, to uh, fix the break. And in this one, uh, you don't need the donor DNA. So when it happened, uh, this repair system stick the break together. I mean, the both sides of break DNA together without any pattern to repair. But in the HDR, the repair system use a pattern to fix and repair the break. Uh, there is three well-known technologies for genome editing: ZFN, TALENs, and also CRISPR. And uh, the reasons for superiority of the technology, I mean, um, a way that found which one is more better than the other one is high efficiency, low cost, time saving, and being stable and inheritable and containing.
Excuse me, sir, to interrupt you. Could you please change the slides, because it's freeze on the first slide and it's present. It's not
Really?
presenter view. Yes.
Thank you for letting me know.
Yeah, we all talking in chat, but it seems you haven't
Oh
seen that,
yeah,
right?
it's
Sorry to interrupt your talk.
no, it's okay. It's very, I appreciate it.
Thank you.
And yes, because when I
It's on
presenting.
PowerPoint presentation. It's not presenter view. I mean, it's a presenter view,
Yes,
not a
yes,
presentation
yes.
mode. Yeah, you should. It should. I mean, you should make
Yeah,
slide
yeah.
bigger on a presenter.
I mean, no problem. If you have any questions, please open your microphone and ask me. Okay.
Thank you.
Thank you for Miss Monday. I'm right. Yeah.
Yes, thank you.
Yeah. And okay. So I try to continue from this one. Could you please give me feedback that you can you see? I mean, the slide.
We can see the slide genome editing, but it's a PowerPoint presentation. I mean, it's not a presenter view. You should click downside. The right side is the presenter view. Then whenever you're talking and change the slide, then we are able to see. Now we can see the PowerPoint,
Okay. Okay.
software. Do you know what I mean?
Yes.
Good.
Is it okay?
Thank you.
Okay.
And it doesn't change while you're talking.
Okay, cool. Thank you for your.
Thank you.
Yes, it was really helpful to me. Okay, so okay. Let me start again from this slide, but because it is more important than the previous one. I talked about it. Yep. And now I'm talking about the mechanism of the genome editing systems. The first was creating a DNA break and then repair the break with two main DNA repair in cells, HDR and NHEJ. This one is HDR. In HDR, you need a pattern, DNA pattern, to repair or fix the break. But in NHEJ, you don't need the pattern donor DNA in editing. So both sides of the break can stick together to fix the break. And okay.
Yeah, we can see slide number 2 now, genome editing.
Really?
Yes. It doesn't change.
It doesn't change.
No.
Oh my goodness. What's wrong?
You go down. Sorry, I mean on the PowerPoint. Go down the right side. Yes, click on this.
Yes.
Apparently you're using 2 monitors.
No, just one.
Okay, but it doesn't come up with the presenter with the slideshow.
Mm. No.
You're using one screen?
Yes.
Okay. And when you click on screen, slideshow, you can see the slideshow view?
Yes.
Oh, sorry, but we cannot. Actually, I
Yes.
cannot. I don't know about
Yes.
the
Yes.
others, but we cannot. It's just
That's
on
the
the
reason
slide too.
that, I mean, confusing to me.
Oh,
That's what's happened. Is anyone here from Parvaz School to help me? این رو شما میدونید چی کار باید کرد؟ تا من پرزنتیشنی که میزنم و شروع میکنم به تغییر اسلاید ها، گویا دوستان نمیبینن. اوکی.
نه وضوحش خوبه، یعنی میتونیم اسلاید رو ببینیم، فقط اگر زمانی که دارین صحبت میکنین همزمان اسلاید ها رو هم عوض بکنین، یعنی بدون در جریان باشین که اسلاید ها عوض نمیشه مگر اینکه شما خودتون دونه دونه انتخابشون بکنین که ما ببینیم شما در مورد چی دارین صحبت میکنین. الان مثلا ما اسلاید شماره 4 رو داریم میبینیم. همزمان که صحبت میکنین اگه خودتون با حالا کیبورد یا ماوس بیارین روی اسلاید بعدی فکر کنم خوب باشه.
اوکی. مرسی از همه. So let's switch to English. Yeah, our Persian is really sweet to me. Okay. The slide. That is HDR. That is NHEJ. In HDR, you need a pattern, a DNA pattern, DNA donor to fix and repair the break. But in NHEJ, you don't need it. Yes, the technology for genome editing are ZFN, Talent, CRISPR, and also the reason or the criteria for classifying the technology according to the abilities of the technology are high efficiency, low cost, time saving, and being stable and containing inheritable modification, and also easy to use. The first technology that I want to have a look, zinc finger nuclease or ZFNs. The system was found by Miller in 1985. He found it in Xenopus, African frog. The core of the system is fork1 endonucleases, and also containing 3 modules with zinc. There is 2 main domains, and the system. First one is cleavage side, and the second is detecting side. And the picture is the endonuclease. Okay. When the system goes to edit a fragment of DNA, as you see, the modules bind to 9 nucleides. There's the binding site, and also, I mean, the cleavage site, as where that the endonuclease stick to the DNA and cut it. The next one is talons. That is an abbreviation for transcription activator like effector nucleases. The system was found in Xanthomonas as a bacteria, as a plant pathogenic bacteria. You know, 3 main parts. The system contains 3 main parts: N-terminal region, C-terminal region, that create two different repeats variable di residues, and also the DNA binding region that is the core of the system. You can see what happened in the editing by talons. As I told you before in the last slide, you can see the talon binding site here and here, and also the core, that it's the endonuclease, the cleavage. I mean, to cleave the DNA. This is, I mean, the RVD, or repeat variable diresidue, that you can find them here. The talon system is so big. It's about the whole system. I mean, it's about 5 kb. So it means it's a huge DNA fragment when you want to transfer it into the cell to edit the DNA. Okay, the main system and so useful and common system is CRISPR-Cas9. That it is an abbreviation for a long phrase: cluster regularly interspaced short palindromic repeats, and the Cas as an endonuclease, which is involving the system, is CRISPR-associated enzyme, that is Cas. The system in nature is bacterial and archaeal immune system against phage or bacteriophage infection. And the most approaches that are used in different biological studies is the most common one is CRISPR-Cas9, and CRISPR-CPF1. That today we know the system as CRISPR-Cas12a, and the most new ones are base editing and prime editing that we talk about it. Okay. In the bacteria, when a bacteria, is infected by a phage, the first, phase in the infection and activating CRISPR system is that the DNA or RNA genome of the virus detected by the system and activate the system to express the pre-cRNA that is a big RNA to then each one that is containing in a spacer and a repeat region cut and they can assemble by Cas9 in the nuclease to find another virus infection. So this is, the locus works as a memory for bacteria to detect viral genomes in future and also in the future generation. There are two main groups in CRISPR editing system that categorize according to involving factor. Group 1, most frequent in the bacteria, and also multi-factor DNA breaking system. It's hard to use and also make large deletion in target site. In genome editing, we need to make it small breaks in DNA to create, an accurate deletion. Because, for example, when you use it for gene therapy, you need that remove a specific region of the damaged DNA. Okay, so large deletion is not desired to us. Group 2, containing the system, the two main system that we talk about it later, and currently we use it in different studies. And two main reasons that they are more useful and common are, single factor DNA breaking system and also easy to use. CRISPR-Cas9 system, as the most common CRISPR system among researchers, and you can find many studies that use the system to edit genome. The main components of the system is cRNA and tracer RNA, which is, according to them, we make sgRNA or guide RNA, small guide RNA. That is about, I mean, 20 base pair RNA, and also protease spacer adjust motif that we noun it as PAM. PAM, you can find the PAM sequence here. It's really short. And here is the main structure of the sgRNA. You can find here each part of the structure. And here is Cas9. Okay. That is the whole picture, that what happened in CRISPR-Cas9 editing. When the whole complex is assembled, the sgRNA found, PAM sequences here, and the next is the cleaving, the DNA, the target DNA. It's about 3 or 5 nucleotides after PAM site. And after creating the break, 2 main repair system can fix it by making deletion and also insertion. That usually HDR works for insertion and NHEJ works for deletion. And about CRISPR CPF1 or Cas12a, here we have 2 main components, cRNA and also CPF1, or Cas12a as the enzyme. One of the most important item of the system is a bit longer PAM site. You can see here, and also I think it's better to find the sequence. And 2 most useful and common CPF1 is LBCPF1 and also ASCPF1. They can edit the target DNA more precisely than Cas9, and the longer PAM site can improve deletion metagenesis and helps HDR to be more efficient and make a sticky end that is enhance HDR efficiency too. Okay, let's have a look to CPF1 enzyme. The enzyme is multifunctional. The enzyme not only cut the DNA, but also it's involving in the cRNA processing. So it helps researchers to use it confidently, because when you design sgRNA, it's a bit, you need to focus more on the RNA secondary structure, and but in CPF1, you can be, you have more convenience to design the sgRNA, and so it makes the enzyme more popular in the last years. However, there are some limitations in both CRISPR systems. The first is limitation in PAM targeting range. Although PAM sites of Cas12a or CPF1 is a bit longer than Cas9, however, it's not something sufficient. So the first limitation can cause the second one. The short PAM site can increase the rate of off-target effect. What is off-target effect? Off-target effect is that the system, the CRISPR system, sometimes cannot be specific and maybe target some undesired region in the DNA. And so what's the solutions? What are the solutions? The first one is enzyme engineering. You can see some Cas in the nuclease here that's engineered to be more specific with improved PAM sites. And also, you can use the ortholix test in the other CRISPR base. The approach can create an irreversible change in a single base without break in a double-strand DNA. Usually, we change cytosine and adenine to I forget the name. Sorry. Change C to T and A adenine to guanine. I think tyrosine. Yeah. And what are the components of the systems of the both systems? For CBE, cytosine-based editing, is cytosine deaminase and also UGC I, that is uracil, uracil glycolase. And but for ABE, you just need adenosine deaminase, and it's really more helpful and useful for you as a researcher that you just change one nucleotide to make a mutation, and efficient for gene therapy of some human diseases, just to change one nucleotide to fix it, to treat the disorder. Base editing is actually a dual base editing system. However, there are some limitations, such as low efficiency byproducts of targets and mutagenesis in genome scale and also transcriptomic scale. And the solution could be using engineered ABES or RNA-based editing, and also redesigning CBES. The last approach is prime editing. Really powerful in editing and making mutations. This system, prime editing, can make 12 different mutations, such as base changing, precise deletion, and also insertion. The components of the system are N-Cas9, which is Nikkei's Cas9, and also a reverse transcriptase that we usually use in RT-PCR, and also a specific guide RNA that which is known as prime editing guide RNA, or pegRNA. And the pegRNA contains sgRNA in 5' end and a primer in the 3' end. And the primer the primer also includes linker and primer for mutagenesis. Okay, I know that the prime editing is a bit difficult to understand because there are many components and maybe a bit confusing at how it works. So let's take a look to the picture that at the first stage, a nick is created in a PAM strait, and then the primer binding site is hybridizated to pamestrin. And the third stage, the reverse transcriptase, replicate, yes, polymerase, the strength in the break, and after that, the cleavage process is completed, and after in the last phase, we can see the ligation and mismatch repair. Let's talk about delivery tools. The tools are really target mediate. That's, what's your goal for using the CRISPR system, CRISPR technology? In research, you can use micro-injection and also electroporation. But in treatment, viral vectors, particularly lentivirus and retrovirus, work for you. But also you can use liposomes and nanoparticles. And liposomes and nanoparticles could be more effective because of when you use the virus, maybe you can maybe you face with immune response. The response to that immune system can show against viruses. In plant science, we usually use gene-gone and agrobacterium-mediated vectors. However, we have some viral vectors, but I've never used them. Okay. The main limitation in the CRISPR technology include off targets that you can fix the problem with designing a precise sRNA. And the second one is museism. That is, it means you have different cells. Some of them are edited, but the other one are still wild. It's not really something desired for you, particularly in plant science. It's not, it's a failure. What are the solutions? You need to accelerating editing process and shortening Cas9 longevity associated with embryo splitting. This is more useful for human and animal cells. And the last one is germline modification, that it is not recommended for animal and human cells because you're not allowed to do it. But in plants, yeah, it's okay. The last one is environmental concerns. That so in all, usually when you are working with recombinant DNAs, you need to follow. You must follow biosafety protocols. That's the references. Thank you so much for your kind attention. You can find here some of my publications on CRISPR and genome editing. Thank you. Just please accept my apologies for my English, because I'm living in New Zealand, and I try to follow American accent, something that is familiar to Iranians, but maybe I have some mistakes. So I'm sorry about that. And also, here is 11:17 p.m. Yeah, I'm so tired, and tomorrow I should go to university, to the lab, doing many lab work. So, however, I'm ready to answer your question if you have any question. No worries, no worries. You're welcome. No question? No worries, you're welcome. Such a terrible silence. Everyone goes out. Do you have any question? Guys, I'm here for your question. You're welcome. No worries, you're welcome anytime. Oh, it's okay if you want. You're free to leave the meeting if you don't have any question or anything to ask. And I'm personally happy to collaborate with you if you want to have any publications on CRISPR technologies. And I know that Parvaz School can support the publication, and they are open to receive your requests for collaborating in publications, particularly Dr. Jalili, my dear friend.
Many thanks, doctor. That was so great. Many thanks.
You're welcome. Anytime. Thank you so much for your time and the opportunity for the presentation, Dr. Jalili. Thank you. Okay, guys, I think that there's no questions, so have a good afternoon and enjoy your Sunday. I should go to bed for tomorrow. Yeah. So, okay. Goodbye. Have a nice day. Thank you, Mr. Mehdi, but it's night here. Okay. Okay, I stay here for even last one. Maybe someone has a question. Okay, guys, I hope that you enjoyed the meeting, the workshop, and Miss Eftekhari, I think. Do you want to say anything? Sorry. Okay, cool. Thank you so much for the opportunity. Have a good day, guys. Bye.